Hi, everybody, and welcome back to this week's lecture. And what we're going to be looking at today is why and how DNA is going to replicate itself. So, our learning objectives are going to be to explain the processes when it comes to DNA replication. And then we're also going to describe and look at exactly what happens when there are errors in DNA replication and how these can lead to specific mutations and exactly what type of mutations they are and how those mutations can happen. So to start things off, what we're going to do is we're going to take an overview of DNA replication itself, and we're going to be specifically looking at this type of model here known as our semi-conservative. So to kind of summarize this up, semi means kind of, conservative means we're kind of keeping some pieces. So in this case, semi-conservative, meaning we're going to have some new and some old pieces. And what you can see here is this is going to be our original piece of DNA right here. And then you'll notice that again, we're starting off with one piece, and then we're eventually going to split apart and we're going to end up with two new pieces. Now, this is going to be known as our semi-conservative because again, what we're doing is we're taking our original strand here and we're going to open it up, or in this case, unzip it. And then we're going to go ahead and find free-floating nucleotides to connect to remember A goes with T, C goes with G. So remember apples and trees, cars and garages. And then they're going to go ahead and bind or find their complementary set pieces together in order to create our new piece of DNA. Now, one important thing is we're starting off with this original piece and we're ending up, although with two new pieces, both are going to be identical to our original piece. And the reason why that's going to be so important is when we go through DNA replication, we're typically trying to either grow or repair something. So it's vitally important that whatever we're trying to do, growth or repair, we are producing more of the original pieces. So again, great examples, let's say you got a paper cut and you were trying to repair that area. You would go through DNA replication, but again, you want to produce more skin cells where skin cells need to be. You don't want to produce any other type of cells because that's going to be very bad. Our body, although as complex and as amazing as it is, it likes to keep its specific things in its specific areas. So again, whenever we're going through DNA replication, we are essentially duplicating or making similar pieces for whatever we're trying to do, whether it be growth or repair. So again, you can see over here in step one, we have two strands of the parental DNA, and those are going to be in blue. Again, our original piece. Each strand acts as a template for assembly for a complementary strand. In this case, you can see in the green parts right here, using supply of free DNA nucleotides. So what that kind of means is, again, we're starting off with our original piece here. It's going to go ahead and open up and unzip. We're going to find our free-floating nucleotides, which means these pieces here. And we're going to use our blue as our template in order to find the complementary sets to build the other half or the other strand. So DNA nucleotides line up along the template strand following the base pairing rule. Again, this is our base pairing rule here. A is going to go with T, and then C is going to go with G. And then enzymes link the nucleotides to form new DNA strands. So they're going to go ahead and connect all these pieces together to kind of lock them in. So now two new DNA double helicases are created and are identical to the parental DNA helix. New strands are referred to as daughter strands. However, no gender should be inferred from this name. So basically what this means is now we're going to go ahead and lock these in. We're again starting with our original piece. It's going to unzip. So we have one half that's going to be old, one half that's going to be new. And then once these are locked in, this is going to be known as our parental, our original piece, and this is going to be known as our daughter. And basically all that basically means is that's our new piece. So again, this process is known as our semi-conservative DNA because after unzipping our original piece, we have one that's old and then one that is new. So now that we went ahead and looked at that, what we're going to do is we're going to see exactly what DNA replication is. So we know that when we do go through DNA replication, we essentially are starting off with one piece. It's going to unzip and open up, and we're going to have one old piece, one new piece. In order to form the new piece, we have free-floating nucleotides. They're going to go ahead and come in and find their complementary sets. So A is going with T, C is going with G. And once they find each other, they're going to go ahead and lock back in, lock up, reestablish those hydrogen bonds. And then now we end up with our semi-conservative model, one half that's new, one half that is old. So what we're going to do is we're going to really dive into exactly what DNA replication is. And we're going to go and explain this in 1, 2, 3, 4, and 5 steps. So remember, we're starting off with our parental DNA, meaning we're starting off with our original or just one piece. And you'll notice that this kind of resembles a zipper. And the reason why it looks like a zipper is, again, we have those base pairs connected together, but we're going to also have something known as our helicase, which is going to be enzyme that's going to help unwind the parental DNA right here, which you can see in purple. And then we have single-strand binding proteins stabilize the unwound parental DNA. So you can see this right here. And then number 3, the leading strand is synthesized continuously in a 5 to 3' direction with DNA polymerase. So you can see we're starting to build those pieces together. And then the lagging strand is synthesized discontinuously. Primase synthesizes a short RNA primer, which is extended by the DNA polymerase to form an Ozazaki fragment. So in this case, this is going to be built continuously down this way. And this one is going to be built this way. And the reason why this is known as the leading is because it's going to go ahead and be built continuously. And this is the lagging because it's discontinuously, meaning we're going to have one part starting to go, stop, go, stop. And we're going to need these portions in order to kind of build and fill those gaps. And then finally, after the RNA primer is replaced by DNA by the other DNA polymerase, not shown, DNA ligase joins the Ozaki fragments in the growing strand. So then that's going to kind of build everything together. Now, I know what you're thinking. That seems like a lot. That is a little bit confusing. But what we're going to do is we're going to go ahead and look at those step-by-step portions when it comes to DNA replication. And then I'll go ahead and try to summarize everything together really, really quickly as kind of like an overview in order to help kind of give you a good synopsis of what DNA replication is. So looking at this over here, we have number 1, a replication fork is formed when helicase, which was shown in purple, is going to separate the DNA strands at the origin of replication. So going back over here, you can see we have those portions. And again, they're unzipping or opening that up. And basically what it means whenever we're unzipping something is we're breaking those hydrogen bonds to separate out the bases. So if we had, let's say, an A and a T here, it's connected by the hydrogen bond. We have that broken apart, and it's going to kind of open up. So what's going to happen is a single strand binding protein, which was shown in red, binds to the single strand DNA to prevent the helix from reforming. Because again, we don't want those to come back together. The main thing is what we're trying to do is we're taking our original piece of DNA and we're trying to open it up. Because again, when we go through replication, we're trying to, again, replicate or make a duplicate of something. So in order to do that, we're using one half of the DNA as a template in order to build the other end, which we will end up with 2 identical pieces. So primase, which was the green box, is going to synthesize an RNA primer, which was going to be in pink, and the DNA polymerase uses the primer to synthesize a daughter DNA strand, which was in light blue. So again, basically what this is meaning is we're synthesizing and we're trying to go ahead and find those complementary pieces in order to build that daughter strand, which is going to be that new strand. So the RNA primers are removed once replication of leading and lagging DNA strands are completed. Okay, so again, leading and lagging, the reason why we have those is because one end is going to be built continuously, and the lagging is always going to try to kind of be keeping up with the leading. Again, that's going to be discontinuously. So DNA ligase, which is going to be the blue circle, is going to seal, think about ligase kind of like as glue, is going to seal the gaps between the Ozaki fragments, and it's going to join the fragments into a single piece of DNA. So now we have 2 DNA double helixes formed. Each of the double helix contains a new DNA strand and one old DNA strand. So this is why we have that semi-conservative model, because again, we have one piece that's going to be new and one piece that is going to be old. So ultimately, one DNA double helix will go into one new cell, and the other DNA double helix will go into the second new cell during division. So again, after we have those 2, they're going to divide out, and then each are going to get their own. So really quickly, I'm going to go ahead and kind of summarize or do a quick overview of exactly what all of this means. And then I'm going to go ahead really quickly and draw this over here. Just give me 1 second to erase everything there. Perfect. So to kind of summarize everything up of what kind of DNA replication is we have our original piece of DNA, right? So we have our DNA double helix right here, just like that, right? And what we're trying to do is we're trying to double this, right? We want to produce another piece of this because again, we're trying to use this for growth and repair. We want to have another exact and identical copy. So the way we do this is we're going to go ahead and we're going to start to unzip our DNA, just like this. So what's going to essentially happen is we have that enzyme, it's going to go ahead and start breaking apart those hydrogen bonds in order to open this up. Once we open this up, because again, the idea or the point of why we're doing this is because we need one end here, so one end here, in order to use as a template to make a new piece. So remember, we have our old strand right here, and we're eventually going to build a new strand somewhere around here. So let's say that we have an A, T, T, and C. Over here, T, A, and G. So now that we've broken this up, what's going to happen is we're going to be looking for free-floating nucleotides like this, and they're going to go ahead and come in, and they're going to be placed in, and we're going to try to connect these back together and solidify them, so then we end up with a second piece. So what we're going to do is we're going to look for new pieces here. So I'm going to go ahead and do that in a different color. Let's go ahead and do that in blue. And we're going to find our complementary sets or pieces. So our A goes with T, T goes with A, T goes with A, and then C is going to go with G, just like that. And then again, we're going to do the exact same thing over here. In this other piece, we're going to look for an A right here. We have a T, T, and then we have a C. So what you should have noticed is between these two, you can see that when we add our free-floating nucleotides, they're going to look exactly identical with each other. Because again, we've unzipped this right down the middle. We're taking our free-floating nucleotides, adding them inside, and then again, we should have our identical piece. So after, again, we unzip them and we add our free-floating nucleotides together, then what's going to happen is we're going to go ahead and kind of lock everything in. We're going to glue it together, and essentially we're going to reestablish those hydrogen bonds. So what's going to happen now is we're going to have one piece of DNA over here, just like this. We're going to have another piece of DNA over here, just like this as well. And then I'm going to go ahead and add in the base pairs to kind of show you what that looks like. So we have A goes with T, 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 C. This would be A, and then G. And then over here, we would have the exact same thing. So A, T, T, C, T, A, G. So you can see that we originally started with, again, our original piece of DNA. We unwound that, we went ahead and unzipped that, and we separated everything out. Remember, we're breaking apart those hydrogen bonds. And then we have, again, our old strands here, old. We're trying to create our new strands in the middle. So what's going to happen is we're going to take our free-floating nucleotides, going to come back in, float in here, add them together. We're going to seal them, set them in, and then we're going to end up with two of these pieces over here. Now, going back to here, one major thing we need to remember is we do have, again, those leading strands and those lagging strands. So remember, leading strand is continuous. So it goes from our 5' to 3' direction. And you can see, again, it's going to be our continuous piece going all the way down. We're going to use our DNA polymerase to kind of place those free-floating nucleotides as we're building that new strand down. And then over here, we have our lagging strand because it's going the opposite direction. So because it's going the opposite direction, we need to go ahead and build it. And there are going to be, again, these kind of little spaces in between. So it's always trying to kind of play catch up to our leading strand. Okay, so again, the lagging strand is synthesized discontinuously, so just not all in one piece. Primus synthesizes a short RNA primer, which is extended by our DNA polymerase to form our Ozosaki fragments. Okay, and then after the RNA primer is replaced by DNA by another DNA polymerase, not shown, DNA ligase is going to join the fragments together, to grow the strand. So again, you can see we have our ligase here, and we're going to be adding those together in order to continuously build or discontinuously build our DNA strand this way. So again, this is the entire process of our DNA replication. We have something unzip it, we have our free-flowing nucleotides come in, we have things that are going to be placing them, we have the reestablishment of our hydrogen bonds in order to lock everything together. We have one part that's going to be leading, which means it's going to be built continuously in one direction, versus our lagging strand, it's going to be built discontinuously, meaning it's not going to be all one singular piece, but it will be made into one singular piece, utilizing all of our different enzymes in order to kind of do that. Again, you can see that our overall direction of our replication is going to be, again, from this way over to this way. So again, that's why this portion is known as our lagging strand. And this portion, because it's following that overall direction, which is going to be known as our leading strand. So I know that this was a lot of information to kind of take in all at once. I do highly suggest that if you are kind of confused, I do have lots of supplementary material in order to kind of show you in a much better and graphical way as to how this kind of works. There is going to be Amoeba Sisters video, which I think does a fantastic job of specifically explaining what's going on with those different pieces when it comes to DNA replication. As always, Dr. Kat's videos are available as well, and I do have simulations and I do have games that should kind of help you get a better grasp or an idea of exactly kind of what I'm talking about. And as always, the textbook does a fantastic job as well of kind of explaining all of that. So again, that was lots of information. We're going to go ahead and move on now to errors in DNA replication. So as you know, whenever we're going through replication like that, there are going to be some errors sometimes. So replication is not always perfect. Errors definitely can occur. And this is when we have certain mutations happen because, again, there is going to be a mistranscription translation, or there's going to be some sort of issue when going through replication. So DNA polymerase inserting wrong bases can happen sometimes. Again, this is the part that's going to help those free-flowing nucleotides get placed where they need to. And there are some times where they just don't match up. So uncorrected mistakes can lead to serious consequences such as cancer. Again, because we want the DNA to look exactly the same when we replicate it, again, that's vitally important. When there are those mistakes, we do have those mutations, and sometimes they can be advantageous or they can be good, but most times they're not going to be great. They can be negative. So we do have certain mechanisms that are going to repair that. So you can see over here, we do have a mismatch of bases detected in our replication. Again, we have our daughter strands that are being built, and we have our blue, which are going to be original. But again, mismatch repaired proteins, detect, remove the bases, fill the gap with the correct bases. You can see here, our G is trying to bind with T, and we know that that cannot work out because A is supposed to go with T, and then C is going to go with G. But again, we can find that, and they can go ahead and replace it. So in rare cases, mistakes are not corrected, leading to permanent mutations in the DNA. In other cases, repair enzymes are themselves mutated or defective. So again, there are going to be cases where these repair enzymes will not be able to fix this, and we do have a mutation, which again can change, alter the DNA permanently. So let's go ahead and take a look at some of those uncorrected replication errors, which are going to result in our mutations. So replication, again, is not a perfect process. Errors do occur, and if uncorrected, can produce mutations. We are going to be looking at two specific types. In this case, our categories are going to be point mutations as well as frame shift mutations. So looking at this over here, when we deal with our point mutations, we have three different types. We have our silent mutations, we have our missense mutations, and then we have our nonsense mutations. So in this case, what a silent mutation is going to be is there is going to be a change or an effect in that DNA, but as luck would have it, it's going to code for the exact same protein. So even though there is a mutation, it doesn't affect it in any significant way. You can see over here that our VAL right here, amino acid, in this case, it should have been G, T, and A, but because we had a mutation right here, and that A became a T, we now have GTT, right over here. But again, that's going to code for the same protein. So although there is a mutation, it is not going to affect it in any way, shape, or form because we're still coding or getting the exact protein that we want. or amino acid. So that's why it's known as silent, has no effect on the protein sequence. So the second type is going to be a missense, and it's going to result in amino acid substitution. So what that means is just like if you have a substitute teacher, it's going to be a change in that specific thing. Looking over here, we're supposed to have SCR, VAL, PRO, and TYR as our amino acids. But you can see right over here, SCR, SCR, VAL, VAL, our PRO has now been substituted with THR. And the reason why is because you can see we have this specific point that's causing that mutation. That's point mutation, one specific position. So that A is going to be changed. I'm sorry, it's going to change to an A. So instead of CCC, now we have ACC. So again, we're changing this one letter, but in this case, this person wasn't lucky to experience a silent mutation. They're having a missense because they have a substitution in their amino acid. So again, it's changing that pro to our THR. Our next is going to be nonsense. So substitution substitutes a stop codon for an amino acid. So in this case, you can see we have SCR, VAL, PRO, and THR. In this case, you can see that our TYR has been substituted for a stop. And this is essentially what's happening is this is telling the sequence to completely stop. We have produced our polypeptide. We're done. We don't need to continue. And this can be a premature stop because we have, again, that one change in that one letter. So instead of it being a TAC, it's TAG, causing this to stop, meaning we're terminating it at this point, and we are not continuing or making more or adding more amino acids. So those are going to be all point shift mutations. Now, when we have a frame shift mutation, essentially what happens here is we can either have an insertion, so an addition of a letter, or we can have a deletion, something taking away. So again, it's going to cause or shift the way we read it, again, changing our amino acid sequence. So over here, you can see we have SCR, VAL, PRO, TRY. But in this case, you can see that we have an addition, right? So we have A, G, C, C. So far, so good. T or G, T, A, G, but it looks like something has happened over here. Now we have C, 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 T, A, C, T, T. So what has happened over here is we have a frame shift, again, shifting where we're reading it. And because we're shifting everything, you can see right here, instead of PRO and TRY, we have LEU and LEU. Because again, we have shifted or changed the way we read this. So again, it's going to cause a completely different polypeptide to be produced. So again, replication is not perfect process, and errors do occur. So mutations are changes in the nucleotide sequence of our DNA strands, and the mutations can lead to changes in the protein sequences encoding the DNA. So just like that, that is going to conclude this lecture. Again, there was lots and lots of information given out relatively quickly. So again, one of the things I highly suggest you do and implore you to do is to go ahead and check out the supplementary material as well as checking out Dr. Katz's videos, because again, it's going to give you a lot of different perspectives and some different explanations as to how DNA replication is going to work. Again, I also highly suggest you check out the games and simulation sections in order to kind of help you understand DNA replication a little bit better. So just as always, there's going to be a test your knowledge. I'm going to go ahead and give everybody about 5 seconds to go ahead and see if you can answer questions 1 through 4, and then we'll go ahead and check out the answers. All right, welcome back. And what we're going to do is go ahead and check this out. So looking at this over here, in number 1, you can say DNA replication is described as a semi-conservative. The reason why is because it's going to be 1/2 old and then 1/2 new. Because again, whenever we're creating or going through DNA replication, we're using an old piece as a template, and then we're going to be creating a brand new strand. So again, we're using a parent one in order to produce a daughter one. Normal DNA replication occurs when or prior to cell division only. And number 3, Circle which of the following types are errors that can occur in DNA replication. So remember, we have all of those different types of mutations. We have our point, and then we also have our frame shift. So again, we can see we have our missense, silent, frame shift, and nonsense. Those are all going to be different types of mutations whenever we're looking at this. Nonsense mutations are going to insert, delete a nucleotide, again, resulting in an insertion of a stop codon. And then a silent mutation is going to change a nucleotide, but has no effect on the amino acid sequence. All right, and just like that, that is going to conclude our lecture for this week. So again, as always, if you do have any questions, comments, concerns, please feel free to reach out. And I look forward to seeing everybody in the next lecture.